Thank you very much for the invitation. I'm not actually a permafrost specialist, so it's a great honor to be here, but we very often in my group work with the permafrost preserved material because it's, the environment is simply so good for long-term preservation of biomolecules, which is really what my group is working on. So we've used permafrost material to address a number of questions. We used it when we sequenced, in 2010, the first ancient human genome. In this case, it was a piece of hair from Greenland, from Western Greenland, originally found in permafrost settings. And we could use this ancient genome from this 4,000-year-old individual to reveal that there had been, in fact, 2 separate migrations of humans into the New World Arctic. So around 5,000 to 6,000 years ago, we had a migration of what is known as Paleoecological, getting into Alaska, Canada, and all the way into Greenland. And then, you know, a few thousand years later, this was followed by the migration of the Inuit people that you can say whose descendants are today occupying these areas, and resulted really in a replacement, complete replacement of this first group of people. So the last Paleoeskimos died out about 700 years ago, not later than that. But they are more closely related to peoples in Siberia than that they are to present-day Native Americans or Inuit people. We also use permafrost material when we sequenced the genome of a 24,000-year-old child from close to Lake Baikal in Siberia. And this is an area that is today occupied by people of East Asian descent. And therefore, of course, we were heavily surprised when we realized, oh my God, what's happening? It's the bad spirit of the child. Okay. Well, okay, it looks fine here, so I'm not sure what's going on, but I'll try to continue even without the slides. But what was really surprising here was that this individual is not closely related to people of East Asian descent. It actually is closely related to people of Northern European descent as well as Native American descent. So we were basically sitting with a kind of, you can say, hybrid between, genetically speaking, a European and a Native American. And I always learned in school that Native Americans is a group of East Asian people crossing the Bering Strait, getting into North America, and there in isolation developing into Native Americans. Well, it didn't exactly fit that picture. And in fact, we could see that all Native Americans today, about 1/3 of their genome, this is a lot, is actually coming from this kind of, you can call it proto-European or whatever individual. So in other words, it seems like Native Americans are really, genetically speaking, composed by the meeting of 2 distinct groups of people, one that is related to, you can say, Europeans, and another group which is related to Asians, and the kind of the child of that meeting becomes, genetically speaking, Native Americans. We also, if we could get at some point some slides up here, but we also used the permafrost material when we sequenced the oldest genome that has been mapped to date, namely that of a 700,000-year-old horse up from Alaska. I did that together with Dwayne Froese, who is also here in the audience. And this was an amazing discovery in the sense that you could actually start looking at how has horse evolution taking place through time, what kind of adaptations, what kind of selection was going on across, you know, from 700,000 years ago all the way up to today. And we could also use this old permafrost preserved genome to address a very fundamental question, conservation biology. Oh, wow, perfect. Here's the child. Oh, here's half the horse. But you know, the Przewalski horse that you can see part of here is believed to be really the only true surviving wild horse. So all other wild horses are actually domestic horses that has then secondary gone into the wild, except the Przewalski horse. The problem, however, has been that it was on the brink of extinction in the 1930s, 40s. There was like 40 or 50 individuals left. And of course, for thousands of years, it has been surrounded by domestic horses. It's found in Mongolia. So the question was there really any truly full-blooded wild Przewalski horse left? And the problem is because you have nothing else to compare to, right? All the other horses are domestic horses, so you don't know what does a wild horse actually look like, genetically speaking. But I can guarantee you that the 700,000-year-old horse was a truly wild horse. And we could actually compare, and we could see, yes, there is in fact still individuals of the Przewalski horse that doesn't contain domestic genetic signatures. And this is, of course, where you should put your conservation efforts. Anyways, actually, this was just the introduction. I don't know how I'm doing in time here, because I wanted to talk to you about something slightly different. And now my clock stopped, so I have no idea where I am, but you'll just have to stop me. Oh, there's one down there somewhere. Okay, so I think what I want to talk to you about is what we know as environmental DNA, because I think that will probably be the most interesting for you guys. So during my PhD, together with a guy called Anders Hansen, we discovered back in 2003 that it's actually possible, with just a few grams of sediments to obtain DNA sequences from a variety of the mammals and the plants that were living in that region when that soil, so to speak, was surface soil. And we actually used permafrost in our original study. So we took permafrost cores that was provided by David Gidishinsky, who is now died, from Siberia. And from those 2 gram sediments, some of them dating back 20,000 years, we obtained the DNA of woolly mammoth, of bison, horses, reindeer, muskox and lemmings, etc. And also a variety of the plants. And this was really, you can say, amazing in the sense, right, that at this time you can say what was available to understand the ecological past. Well, we had pollen diagrams and we had macrofossils. And then if we were lucky, in a certain area, maybe we could find some bone and teeth, and then we could say, well, there has been a reindeer here or whatever. But everything being equal, the chance of, especially outside the permafrost regions, of finding something from animals is not necessarily very high. And all of that you could just get in a few grams of sediments, both in permafrost. There we went back all the way to about 500,000 years. So it's a long time period you could cover, but also for non-frozen sediments as well. And if you want to read a review on this, I think this is a very up-to-date review we did very recently. And when we did this, it came out by chance, basically. We were non-famous. We couldn't get access to any interesting bones and teeth, so we had to find out to do something else. And we thought, what can we do? Well, look at the dirt that everybody throws out, right? And that turned out to be a gold mine. And at this time, we didn't know where's this DNA coming from. We still don't know a lot about it, but we do know something. So it really seems like the DNA you find in the ground is not really mainly coming from pollen grains. So pollen grains isn't the source of the plant DNA. It seems like fine plant rootlets, less than 2 millimeter in diameter, that's constantly being shed, as well as other somatic tissue seems to be the most main source of plant DNA. For animals, it seems to be mainly feces and urine. So with the feces, you're actually releasing, you can say, cells from your intestines that contains, of course, the DNA of the source animal, and the same from the urine bladder. And when these cells are getting into the ground, of course, some of it will survive as, you can say, as tissue, as micro or macro fossils, but some of it will also be lysed. It will be, the cells will be opened, the DNA will be released to the surroundings, and a lot of this will be eaten by bacteria. I mean, they will use it as energy. But because DNA is electrically charged and some soil particles are also electrically charged, some of this DNA will bind to these particles, which will actually reduce the rate of degradation. We have also recently found out, we had a paper, I think last year or the year before, I can't remember, but that bacteria, living bacteria, can actually take up some of this ancient DNA, ancient degraded DNA, and incorporate it into their own genomes. So it means you can actually kind of get genetic signatures of the past, in principle, half a million or a million years ago, being revived into living bacteria. It's pretty remarkable. We don't know what rate this is happening on, but we know it's certainly happening. There's no doubt about that. Okay, so all this DNA from sediments and permafrost, to what extent does it actually reflect the real community? I think this is a very reasonable question to ask yourself. And in order to address this, we have done a number of, you can say, surface studies, right, where we know the vegetation, we know the animal life. If we take soil samples, what do we get? For example, we did a study together with also some people here in the audience up in northern Norway, where we took soil samples, surface samples, in an area where you can say the vegetation had been covered by traditional method, right? Taxonomists running out, saying how many of this tree is then, how many of that, etc. And there is, in fact, a very nice correlation, I think, both qualitatively in terms of what kind of organisms do you cover, but also to a surprising extent, to me surprising at least, quantitatively. It's not perfect, but there is a clear correlation there in terms of how much biomass do you actually get and how much DNA do you get. It's not perfect, but it's not bad either, definitely not. We have also done it with animals. Here we used actually soil samples from zoo parks, because zoo parks, you have all these exotic animals, right, in Denmark, elephants and stuff. And you know exactly the number, when they were introduced, right? And then we took soil samples from these zoo parks, and in fact, to a large extent, it's the similar pattern. There is some places where it doesn't match, and that seems to be related to the animal behavior. So some animals, there was a gazelle, for example, they were always standing on a hill, right? They were never moving. So unless you're taking a sample from that hill, you won't catch it, basically. So there you had to take that into account. Okay, so what can you then use this for? So one of the things that my group has been very interested in is, the extinction and the dynamics of the megafauna, the big-bodied mammals that lived in the northern hemisphere, in the end of the last ice age. Woolly mammoth, bison, horses, etc. And as you probably know, some of you are much more specialists within this area than I am. We know that for the large parts, they got extinct, most of them presumably around the end of the last ice age. And it was like a mass extinction in the sense we are talking about a loss of about 2/3 of these big mammals. And what was the reason for this? This is something that has puzzled science for decades. And there's different hypotheses. Some argue, it's human-based. It's due to humans getting into these regions. These animals have never seen humans before, and they were basically slaughtered one by one, and it happened very quickly, within 1,000 years of first human contact, extinction by blitzkrieg. Other people say, it's ridiculous to think that a few humans with a primitive technology could take out all these animals across both, the Americas, Siberia, Europe. It must be due to climatic changes, change in the vegetation, and then a collapse of, you can say, basically the mammalian ecosystem. And of course, the skeptics here say, how can it be that these animals could survive climate changes in the past? but now, at around the end of the last ice age, what is so special about that event causing the extinction? And then there's even been suggestions about an extraterrestrial impact hitting northern North America 12,900 years ago, causing all kinds of disturbances, wildfires, etc., and creating some climate change events. So we attacked this issue in 2 papers. The first one was more traditional in the sense that we collected basically animal bones from across Europe, Asia, and North America, obtained DNA from these bones and got C14 dates on them. And then we used the DNA to basically reconstruct, you can say, how has the effective population size of these animals changed through time? In other words, how has the number of animals changed through time? We also used the date and the location to do so-called climate niche modeling, where you basically ask yourself if only temperature and precipitation was influencing the distribution of these animals. What would the predicted distribution of these animals have been through different time periods, based on the knowledge you have about climate and the location of these animals, in certain areas. Finally, we revisited the archaeological record, asking, where are humans at the time where you see these animal events? To what extent are humans actually exploring the different groups of these animals? And from this large data set, we were talking more than 100 of individuals from each group. We saw a number of very interesting things. First of all, oh my God, I'm running out of time here. Okay, this has to go really fast. Okay, so first of all, what we can see is that there's a very nice correlation between the climate niche and the changes in population size. So to me, there's no doubt that climate change is a major driver of these animals' population dynamics. In terms of humans, well, in a number of cases, we don't see even a geographical overlap between humans and the animals around the time of extinction. So in some cases, humans can have played a role, I believe, taking out the last ones, but as the main driver, I have a hard time see how that could be humans. And now the last thing and we need to get this one, because this was kind of what really, I hope, would be very interesting for you. So in order to so it seems like climate is really a major driver. What about the climate? Is it because of vegetation changes? And in order to address this question, we basically collected permafrost samples from across Eurasia and North America, and took all the samples that could be C14 dated, so it means covering the last approximately 50,000 years. We also got access to these mummies of megafauna, a frozen woolly rhino or frozen mammoth, and got stomach content from them. And we did DNA investigation on all this. And what can we see? Well, we see something very interesting. So before, you can say between 50 and 30,000 years ago, the diversity of plants in the northern hemisphere up here is much, much richer than today. But it's not dominated by grasses, as has been very often reported in the literature. This is not what we find. We find that it's dominated by forbs, but it's very rich in terms of diversity. Then we get into the last glacial maximum, right? The coldest and driest stage of the last glacial, and we see a massive reduction in diversity, which is really biologically makes a lot of sense. It's not what has been seen pollen-wise, but if you get to really harsh conditions, of course you will see a reduction in species diversity. However, it's still a subset of the diversity you found in the previous period. But then when you get to the Holocene, we see a complete change in the vegetation into the vegetation that we see today. If we then look, so the first lesson here is that even though that the climate, so to speak, bounced back, right, it doesn't mean that the vegetation necessarily will bounce back, right? So in other words, there's also a vegetation history to take into account. If you first have lost something during the last glacial maximum, for example, then when it's getting warmer again, it can be something very different coming in and becoming dominant. Maybe you would think, oh yeah, then we just return to what we had before, right? But no, it's not what it happens. And this is, of course, one of the reasons why you can't just compare. When you talk about the extinction, you can't just compare why didn't they survive the previous climate change period, because the vegetation doesn't have to be the same, even though the temperatures and the precipitation might have been similar. But then when we look at the stomach content of these animals, what seems to be the dominant food source for these megafauna animals is the protein, is these forbs. And see, what is striking is the forbs are not reduced during the last glacial maximum. Where you really see a massive reduction of those is in the Holocene. And this is around the time where most of these animals goes extinct, right? So in my view, I'm sure there's a number of people disagreeing with me, I see, climate and vegetation history as the major driver of these events, and humans, you can say, as secondary in this whole process. And it really tells us that, these climate changes that we are facing now, can really have some pretty severe impacts, right, on our surroundings, and we can get a lot of changes. And now I'm out of time. So thank you very much.